Welcome everybody. My name is Christian Krone. I'm a pharmacist and PhD student, and it's a great pleasure for me to introduce you today to DSC for characterization of nano-based drug delivery systems. Let's start directly with the overview. I will show you today two applications. The first one is DSC analysis of crystallinity variations for nanoparticles prepared with sustainable solvents. And the second application is the simulation of iron oxide nanoparticle degradation detected by DSC. So, let's start with the application number one. Over the last 20 years, we saw an increasing number of PLGA nanoparticle publications for drug delivery. They all have in common that they were prepared by standard formulation methods like the single and double emulsion, nanoprecipitation, salting out, microfluidic technology, or membrane emulsification. All these methods use organic solvents, and these solvents can have some limitations. They can be damaging for the environment, they can be toxic for the human health, and they are very cost intensive to remove and to recycle, especially when it comes to large industrial manufacturing. And therefore we need new preparation techniques which are non-toxic and sustainable. Here we can see the composition of these nanoparticles. We need a drug. We use atorvastatin. It is a cholesterol-lowering drug. It has some anti-inflammatory properties, and it is highly lipophilic. And therefore, we need our nanoparticles to encapsulate this highly lipophilic drug and to bring this drug into the patient. Then we have a polymer. We use PLGA. It is a copolymer of lactide and glycolide, and it is biocompatible, biodegradable, and approved in many applications from the authorities. Then we use a proper solvent to dissolve the drug and the polymer, and therefore we use ethyl acetate as a standard organic solvent. It is partially water miscible, and it has some toxic properties. We also have two sustainable solvents, the Cyrene. It is a cellulose-based solvent. It is water miscible, biodegradable, non-toxic, eco-friendly, and we also use polyethylene glycol 400. It is also sustainable, water miscible. It is non-biodegradable, non-toxic, and eco-friendly. Here we have an overview of our preparation methods. We used three different methods: our standard method with the ethyl acetate, the Cyrene method, and the PEG 400 method. And the big advantages of the sustainable preparation methods in green is that we have a huge reduction of the process time. So when we now prepared our nanoparticles, we characterize them for the physicochemical characteristics. And here we can see that we have comparable results between the preparation methods. We see on the left side the hydrodynamic diameter. That is a measurement for the particle size. We have comparable particle sizes. We have also a very narrow particle size distribution and a negative theta potential, which is a measurement for the stability of the particles in dispersion. We also checked the morphology of the nanoparticles. We can see for all preparation methods that we have spherical shaped nanoparticles with a smooth surface. Now we come to the interesting part of our study. We investigated our nanoparticles by DSC, and we looked at the class transition temperature and the enthalpy. And what we were found was that we have a comparable class transition temperature as well as melting point enthalpy between the pure PLG polymer, the physical mixture of PLGA and atorvastatin, the emulsion method and the Cyrene method, but when we use our PEC 400 method, we have a strong decrease of the glass transition temperature as well as the melting point enthalpy. When we then use another polymer, the PEC PLGA, where PEC is covalently bound to the PLGA, then we see the same strong decrease of the glass transition temperature and the enthalpy. And therefore, we can say that the nanoparticle crystallinity and glass transition temperature is dependent from the type of the polymer and from the type of the solvent. We also see these plasticizing effects of PEC, whether it is covalently bound to the polymer or it is somehow involved in the preparation process. And therefore, we can say that the DSC is a suitable technique to distinguish between the preparation methods. We also use temperature-dependent AFM measurements to investigate our particles, and what we found was that our particles lose their structural integrity independency of the temperature, and we also saw that this happens in the temperature range of the TG which were obtained by the DSC analysis, and therefore we were able to confirm our DSC results. So the question is now, why is the crystallinity and the glass transition temperature so important for us? The answer is that they have a huge impact on many nanoparticle properties, like the in vitro drug release, as well as the degradation of the nanoparticles. And therefore, we investigated the in vitro drug release, and what we found was that nanoparticles prepared by the PEC 400 method, here in blue, have the fastest drug release of all three methods. And the emulsion method has the slowest drug release. And therefore, we can say that the difference of the nanoparticle crystallinity resulted in a modified in vitro drug release. Now we come to our second DSC application for the simulation of iron oxide nanoparticle degradation detected by DSC. At first, I'd like to give you again an overview about the rationale of iron oxide nanoparticles. They can be used for diagnostic reasons, for MIT investigations or molecular imaging. There are many products on the market, and you can also use it for the therapy, especially for cancer therapy, to induce magnetic hypothermia, for magnetic drug targeting, or hypothermia controlled drug release. Here we have an overview of our magnetic nanoparticles. We always use an iron oxide core consisting of different core materials, and the core is then coated with different shell materials, with natural materials, semi-synthetic materials, synthetic materials, and inorganic materials. All particles have comparable particle sizes, and they differ due to the different shell materials in their particle surface charge. So we have positive, negative, and neutral charged particles. The aim of our study was to simulate the degradation and the stability and the fate of our nanoparticles in the body, and therefore we used simulated body fluid, shortly SBF, at a pH of 7.4. It is highly suitable and able to simulate the biological conditions of the blood plasma and the intracellular environment in terms of salt composition, pH, and osmotic pressure. And therefore it is possible to simulate the environment after and during the injection of our iron oxide nanoparticles and the distribution of the particles. So we can say that the perfect diagnostic magnetic nanoparticles would be very stable in SBF. By DSC analysis, we were now able to monitor the stability of the iron oxide nanoparticles in simulated body fluids, and we were also able to distinguish between the shell material and the core material. So here we see the results for the shell material. Therefore, we performed DSC analysis under nitrogen atmosphere, and we made this before storage in SPF and after 28 days storage in SPF. So we selected 3 different types of nanoparticles with different shell materials. We selected a positive charged branched pi nanoparticle, a neutral charged pegdextran particle, and a negative charged polyglucuronic acid nanoparticle. And what we found was that the shell of the polysaccharide-coated magnetic nanoparticles, so this means the pegdextran and the polyglucuronic acid, was not affected after storage in SPF, because the signal after storage is highly comparable to the signal before storage. And we also found that the shell signal of the branched pi-coated nanoparticles was lost during storage in SPF. Iron oxide nanoparticles can exist in different redox states, which determines the magnetic properties of the nanoparticles and can be oxidized during the storage of the particles, and therefore it is very important for us to investigate these oxidation. The different redox states of the iron oxide can be the magnetite, the mechamide, and the hematite, which has no magnetic properties anymore. So here we can see the results. We investigated pure magnetite and pure mechamide cores with a DSC under oxygen atmosphere, and we can see a black the black line. This is the magnetite, where we see a large peak number 1, and we see the blue line. This is the mechamide, where we see no such a peak number 1, and therefore we concluded that we have a conversion of the magnetite to the mechamide. We also applied a second test cycle to the magnetite, where we see no peaks anymore, and therefore we can say that we have a complete oxidation during the first run. We also see for all nanoparticles with a shell, I show it in a few seconds on the next slide, a second peaks, and with our references here we see no such peaks, and therefore we concluded that a peak number 2, which we see on the next slide, that we have here a conversion of the hematite to the mechamide. Here we can see the results for the shell-coated magnetic nanoparticles. We saw that all the magnetic nanoparticle cores consisted of different oxidation states and conformations, because all these nanoparticles showed a large peak number 1 and number 2. We also saw that that peak number 2 became more prominent during the storage for the magnetite cores. So this means the branched pi coated nanoparticles and the pegdextran coated nanoparticles. In contrast, we didn't see this effect for the polyglucuronic acid coated mechamide nanoparticles. In conclusion, we can say that DSC analysis is able to detect the changes of the core and the shell of the magnetic nanoparticles. In summary, we can say that the DSC is an important tool to distinguish between nanoparticle preparation methods and their effects on the in vitro drug release. And as a second application, it is an important tool to simulate the biological effects of iron oxide nanoparticles. In the end, I'd like to thank my supervisors, Professor Dr. Dagmar Fischer and Professor Dr. Oliver Wertz. I also like to acknowledge my cooperation partners from the PolyTarget project and the Nanobel project. And when you have any questions, you can always contact me under my email address. Thank you very much.